All right, in this lecture, we are going to be talking about cell-to-cell communication. Basically, how do cells talk to each other? This is really important for unicellular organisms in order to be able to talk to other individuals, but it's also important for multicellular organisms to be able to talk to cells within the organism and coordinate responses. Now, we know that there are some universal mechanisms of regulation, and that most of the communication that takes place is through chemical signals. One example of signaling that you're probably familiar with is the idea of flight or fight response. This is where the body is preparing to either stand and fight or run and flee from a threat. This response involves all sorts of physiological changes, like your heart rate increasing, your respiratory rate increasing, your pupils dilating, blood flow to your muscles increasing, all things that are preparing you for this emergent situation. And it's all mediated by a signaling molecule called epinephrine. So we'll look at that more today and in other lectures. Now, one of the ways that we have studied cell signaling is by looking at a species of yeast that has two mating types, an A type and an alpha type. Now, what happens is the A type cells secrete mating factors that are different from the mating factors that the alpha cells secrete. Once those factors connect with their target cell, they set off what's called a signal transduction pathway, which is where a signal from the cell's surface, the mating factor, gets converted into a specific cellular response. And it's almost kind of like a game of telephone, where you have individuals standing next to each other and one person tells the message to the second person, who tells the message to the third person, who tells the message to the fourth person. So imagine if that was taking place, but there was a door in between the first and the second person. They might have to change the way that they're giving the message. Maybe instead of saying it out loud, they'd have to tap it using Morse code or pass a note through the door, something like that. That's the idea. So if we take a look here, we have an A yeast that's secreting A factors and an alpha yeast that's secreting alpha factors. Notice that the alpha cell only has A receptors on its surface, and the A cell only has alpha receptors on its surface. Once those receptors are filled with signaling molecule, it triggers a change inside the cell that allows mating to take place, where the cells fuse together and form a new cell that has genetic material from both the A and the alpha cell. This particular process we won't go into more detail on. It's just giving you an idea of what kind of response you could see. We also think that prokaryotes have evolved similar signaling molecules that then became modified later in eukaryotes. Remember, eukaryotes came around at least 1 billion years after the prokaryotes, So, for example, with bacteria, bacteria exist as individual rod-shaped cells, pictured up here on the left. But if a bunch of signaling molecules are concentrated in one area, the bacteria kind of sense that there's a high level of population density, and they'll start to aggregate. They aggregate and form a spore-forming structure known as a fruiting body. This is what they actually look like, which allows them to spread. So again, kind of a neat example of cells communicating with each other within the prokaryote kingdoms or domains. Now, we can see both local and long-distance signaling primarily accomplished by chemical messengers. As you know from other lectures, animal and plant cells have junctions that allow them to connect directly to the cytoplasm of adjacent cells, like a communicating or gap junction in animal cells, or a plasmodesmosome or plasmodesmata in plant cells. So in these local signaling, sometimes the cells are communicating by direct contact with each other, or cell-cell recognition. They're actually physically touching each other. But in a lot of other cases, animals will use regulators. Now, local regulators are messenger molecules that only travel a short distance. But long-distance signaling uses hormones, which are signaling molecules that travel through the bloodstream or through the vascular structures, and they can travel all over the body. Whether a cell will actually respond to that hormone, for example, whether a cell responds to epinephrine and what its response might be, is totally based on whether or not it has the receptor that's specific to that signal and which receptor it has, because sometimes receptor cell signals have more than one receptor. So here's those gap junctions that allow for direct communication, plasmodesmata. Here we have cell-cell recognition, where the cells are physically in contact with each other, often through the use of glycoproteins on the cell membrane. In this case, this green glycoprotein is hooking up to this purple receptor and setting off a response inside the second cell. We also have local signaling. There are a couple kinds of local signaling. One is called paracrine signaling, where one cell releases signal, it travels through the extracellular fluid and targets cells that are local to it. We also have synaptic signaling, where a neuron releases neurotransmitters, which are just chemical signals, that diffuse across a small space called a synapse. Actually, this whole thing is called a synapse. That space would just be the synaptic cleft. And then they hook up to receptors on their target cell to stimulate a response. For long-distance signaling, we will see endocrine cells release hormones into the bloodstream. They will travel through the bloodstream, and they will likely come in contact with almost all of the cells in the body, but they will only affect the cell if the cell has the appropriate receptor. So let's break down the 3 steps of cell signaling: reception, transduction, and response. And we're going to go through more details on each of these stages. One of the ways this was determined was by a man named Sutherland, who first started to discover how epinephrine could affect different cells. In reception, you have a signaling molecule that binds to a receptor that is often in the cell membrane of the target cell. It can actually be inside the cell as well, depending on the type of molecule. We'll look at that later. Once that signaling molecule binds to the receptor, that will set off a chain reaction of relay molecules that will end up leading to a response in the cell. This portion, this chain reaction that's been set off inside the cell, is known as transduction. Then that can lead to a response, and that response is often related to gene regulation, like making more proteins, or it can even be like building certain structures within inside the cell, changing the shape of the cell, all sorts of things like that. So let's look at reception in more detail. The signaling molecule is generically called a ligand or ligand, and receptors are highly specific to those ligands. So usually what happens is a ligand or signal molecule comes up to a receptor, binds to it, and changes its shape in some way that will then set off that transduction pathway. Most of the signal receptors are plasma membrane proteins, but as I mentioned, some of them are proteins that are inside the cell. It's very dependent on what type of signaling molecule it is. If it's a signal molecule that can pass through the cell membrane, the receptor will be in the cytoplasm. But if it's a polar or water-based signaling molecule, it can't pass through the membrane, so the receptor has to be in the membrane. So let's look at this. Most water-soluble molecules bind to sites on receptors that are in the plasma membrane. And there are 3 main types of receptors we're going to look at: G protein-coupled receptors, receptor tyrosine kinases, and ion channel receptors. And what we're doing right now is we're getting a basic understanding of what these different types of receptors are so we can build on it and build some signal transduction pathways for you to understand. So G protein-coupled receptors, or GPCRs, are the largest family of cell surface receptors, and they are a receptor that work with the help of what's called a G protein, which can act as an on-off switch. When GDP, which is guanosine diphosphate, kind of like adenosine diphosphate or ADP, when GDP is bound to the G protein is inactive. So let's see what that means. Here's our GPCR, right? So here's our receptor. You'll notice that there are these 7 alpha helices of the protein that are in the hydrophobic zone. And then we have hydrophilic portions sitting on the outside of the membrane, including the signal molecule binding site. and hydrophilic portions sitting on the inside as well, the part that will interact with the G protein. So here's what happens. We have a GPCR, and then we have the G protein with GDP bound to it, so it's inactive. Then over here we have an enzyme. We're going to look at examples of those enzymes, but for now let's just know it as an enzyme. Once the signaling molecule activates the receptor, it interacts with the G protein so that the GDP is released and GTP is attached. Now, guanosine triphosphate has more energy available in it, right? Because it's got an extra phosphate bond. That GTP then will interact with the enzyme, and that will trigger a cellular response. After that's happened, the G protein stops interacting. It gets dephosphorylated, right? It loses a phosphate, and it goes back to its inactive form of a G protein attached to GDP. So that's why we call this a GPCR, because it's G protein coupled. It's a receptor that works with a G protein. Now, receptor tyrosine kinases are the second type of receptor for us to talk about, often called RTKs. Their job is to attach phosphates to tyrosines. And that, in general, by the way, is what a kinase does. Kinases attach phosphates to things. Now, what's cool about RTKs is that they can trigger multiple signal transduction pathways at once, and you'll see why when we look at their structure. They are often very responsive to things like growth hormones, so abnormal functioning of RTKs can be associated with lots of different types of cancer. So here we have our RTK proteins. You'll notice there are two proteins separated that are full of tyrosines and have ligand binding sites on the outside of the cell. They are in a form called inactive monomer. It means one monomer of protein is here, one monomer of protein is there. When signaling molecules come in and bind to both sites, it causes the RTK to form a dimer, where the two pieces are connected to each other. And that is its active form. This then activates those tyrosine kinases so that they get phosphorylated with ATP. They're now fully activated and ready to start triggering some pathways. For example, in this case, this one's activating the top protein to trigger one response, and this one's activating another protein to trigger another response. So again, because they have so many sites on them, they can trigger multiple responses at the same time. Now, the third receptor we'll look at is a ligand-gated ion channel. This is where the receptor is a gate, and when the receptor changes shape, it opens or closes the gate. So for example, a signaling molecule could bind to the receptor, and then the gate would allow a specific ion to move through it. This could be a sodium-gated ion channel, a calcium-gated ion channel. So for example, here we have a closed gate. This is our ligand-gated ion channel receptor. A signaling molecule that fits comes in and binds to it, changes its shape, opening the gate so that ions can rush through, which triggers a cellular response. The ligand gets removed and the gate closes back up. We see these a lot with calcium-gated channels, by the way. Now, I mentioned that water-soluble signaling molecules have receptors in the cell membrane, but hydrophobic or nonpolar signaling molecules have their receptors within the cytosol or even the nucleus of the target cell. These are called intracellular receptors. So these would be things like steroids and thyroid hormones that can diffuse through the cell membrane of the cell and go into the cytosol or even the nucleus, bind with their receptor, and trigger a response. Often, this binding of the hormone, like the steroid hormone, which are things like sex hormones, estrogen, testosterone, often they will bind with a receptor and form a transcription factor. As a reminder, transcription is the process of copying a small piece of DNA that has the instructions for building a protein so that onto a piece of RNA so that that RNA can leave the nucleus and help build the protein. So a transcription factor encourages transcription of a particular gene. So here we have testosterone. It's going to diffuse right through the plasma membrane and bind to its receptor protein, forming a hormone receptor complex that will diffuse into the nucleus and act as a transcription factor, encouraging transcription at this site on the DNA, which produces a particular piece of mRNA, which leaves the nucleus to help build a specific protein. So in this case, this testosterone hormone has basically activated the protein synthesis of a particular gene. All right, let's add in some information about transduction, and as we do, we will start building up some pathways. So signal transduction has lots of steps, which helps it to amplify a signal. We'll see an example of this in a moment, but you can have a few signal molecules that produce a very large cellular response. Additionally, having multiple steps in your pathway helps allow for better regulation and coordination of the response, because at each step, that's an opportunity for regulation. Most of the molecules in the signal transduction pathway are proteins, and they sort of act like a series of dominoes where the receptor will activate one protein, that protein activates another, which activates another, which activates another, until we get to the response, okay? And this activation of, when we say activating a protein, we're really referring to changes the shape of the protein in some way. Now, there's two types of enzymes that play a strong role in many of these pathways through what we call a cascade of phosphorylations. You have protein kinases, which phosphorylate things, they add phosphates, and you have protein phosphatases, which remove phosphates, dephosphorylation. So you sort of have this phosphorylate, dephosphorylate, phosphorylate, dephosphorylate pattern, which acts as a molecular switch to turn the activities on or off as required. So here we have our signaling molecule binding to the receptor, which triggers this activated relay molecule to activate protein kinase 1, which activates protein kinase 2, which activates protein kinase 3, which activates some protein that causes the cellular response. So all of these kinases working together, that's our phosphorylation cascade right there, our chain of dominoes of things interacting. Now we also see a phenomenon known as second messengers. The ligand that binds to the receptor is the pathway's first messenger. It's the first signal. But sometimes that first messenger sets off a pathway that leads to the release of a second messenger, which continues the pathway. So second messengers are usually small, water-soluble molecules or ions. They're usually not proteins, and they can spread around through diffusion. Okay, again, we're going to look at an example of this. We see them play a really strong role in GPCR-mediated pathways, as well as those that are initiated by RTKs. So two of the most common second messengers we're going to look at, and these are going to help us build those pathways I mentioned, are cyclic AMP. AMP, by the way, is adenosine monophosphate, one phosphate, unlike ATP, which has 3, and calcium ions. So let's take a quick look at cyclic AMP. It's one of the most widely used second messengers, and it is formed by using an enzyme called adenylyl cyclase. Now, this is an enzyme that exists in the plasma membrane, and it'll convert ATP to cyclic AMP. So here's our ATP. Adenylyl cyclase removes 2 of the phosphates off, and we're left with cyclic AMP. Then there's another enzyme, which we're not going to talk about in class, but it's called phosphodiesterase, that removes, that moves, causes an isomerization of that phosphate group, forming AMP. But the cyclic AMP part is the part that we're interested in. Okay, this is going to be one of our second messengers. Now you have lots of signal molecules that can trigger the formation of cyclic AMP. They are often associated with G proteins and GPCRs, G protein coupled receptors, as well as protein kinases. And usually cyclic AMP will activate a particular kinase, protein kinase A, which will set off that phosphorylation cascade we talked about. So a way to, again, we're going to look at an example of this, but this is important because one of the ways that we regulate cell metabolism is through these G protein systems that basically inhibit adenylyl cyclase from making cyclic AMP unless it's the right time for it to be made. So let's track this signal transduction pathway, and this is one of the ones you should know. You have our first messenger, which is our signaling molecule, like epinephrine, that connects to its GPCR, which activates the G protein, which activates adenylyl cyclase. Adenylyl cyclase then takes ATP and converts it into cyclic AMP. Cyclic AMP is our second messenger that then sets up a phosphorylation cascade with these protein kinases that will lead to a cellular response. For example, in the case of epinephrine, the cellular response might be to break down stores of glycogen in muscles to make glucose available for the emergency that your body is perceiving. So this is a pathway you'd want to be able to explain each step, like I just did. Now, the second big pathway I want to focus on is the IP3 pathway. So here's where we're going to see calcium ions play a second messenger. And one of the reasons that calcium is such a good second messenger is because cells can actually regulate their own concentration of calcium by using pumps and gated channels to store calcium in particular locations. So for example, here we have a pump that is pumping calcium out into the extracellular fluid. So take a look. The cytosol has a relatively low concentration of calcium. The external fluid has a lot of calcium in it, as does the internal spaces in the membranes. So like inside the endoplasmic reticulum, inside the mitochondria. And there are pumps that are maintaining this and allowing this to happen. And they do require energy because we are pushing calcium against its concentration gradient from where it's low to where it's high, from where it's low to where it's high. So signals that get released by signals that come from a transduction pathway can trigger the release of calcium into the cytosol. So for example, opening a channel here so that calcium flows out into the cytosol or flows into the cytosol from the outside of the cell. Okay. So one of the pathways that leads to the release of calcium is the IP3 pathway. We'll also see something called DAG or diacylglycerol play a role. IP3, inositol triphosphate. Those are going to also be second messengers. So this pathway, I really like this next one because it really highlights all of the concepts we've talked about so far. So let's talk through it step by step. Number one, a signaling molecule, which is our first messenger, connects to the GPCR. This activates the G protein, which activates an enzyme. In our previous pathway, the enzyme was adenylyl cyclase. But in this IP3 pathway, the enzyme is called phospholipase C. Phospholipase C cleaves a molecule, meaning cuts it, called PIP2. You don't have to know what PIP2 stands for. It cuts it into two pieces. DAG, which stays up here and becomes part of a different pathway that we're not going to worry about, and then IP3, which is our second messenger. IP3 then is going to travel all the way over to here where you see an IP3 gated calcium channel. That means this calcium channel stays closed unless IP3 binds to it. It binds to it and allows the release of calcium. Calcium is then our next second messenger because this increase of calcium ions is going to trigger the activation of proteins, which will lead to cellular responses. So again, both the cyclic AMP pathway you'd want to know and this IP3 pathway you'd want to be able to explain step by step. Now, the last major piece of all of this is what kind of response are we getting from this signal transduction pathway? Often we call it the output response. Well, you can either have a nuclear or a cytoplasmic response. It's always going to be one or the other, or it could be both, okay? So many signaling pathways will regulate the synthesis of enzymes or other proteins so they can turn genes on and off in the nucleus. We saw that with the testosterone example, right? Where the hormone receptor complex acted as a transcription factor. So for example, this is kind of an interesting one. Growth factor on the outside. Notice that the growth factor can't diffuse into the cell. But what it does is it binds to the receptor, which sets off a phosphorylation cascade, that's our transduction piece, which ends up activating a transcription factor so that it triggers a response of making RNA to build a specific protein. This response that we're seeing here is very similar to what we saw with our testosterone example, The big difference is the growth hormone didn't come in and find a receptor to do it. It set off a long transduction pathway to get it done. Another way that, again, with that cytoplasmic response, so this would be an example of a nuclear response because it's happening inside the nucleus. A cytoplasmic response could be increasing the activity of enzymes. So this is also, by the way, this image is a great example of that phenomenon of amplification that I mentioned, where like one signaling molecule of epinephrine can trigger a massive response. Let's take a look. One molecule of epinephrine binds to a GPCR. That activates 100 G proteins, which activates 100 adenylyl cyclases, which activates 1,000 cyclic AMPs. All the way down, all the way down, all the way down, until we eventually, what we've done with the activation of all of these enzymes is we have converted glycogen into 10 to the 8th molecules of glucose. So one epinephrine made a huge amount of glucose available in the cell. That's amplification. This is also a cytoplasmic response because this is all happening in the cytoplasm. It's not about gene regulation, it's just about breaking down glycogen. Now, one cool thing that we sometimes see, I mean, I think it's cool, that we sometimes see is that the signal transduction pathways can actually lead to a change in the shape of a cell. So let's look at these over here. So here we have a wild type of this organism. Wild type means what we see in the wild. And you'll notice that you have organisms that form these extensions in their cells called shmoos. These shmoos are mating extensions so that these cells can mate with each other. Let's talk through how it works. So there's a mating factor, kind of like what we saw with our yeast example, the alphas and the A's at the very beginning of the lecture. A mating factor gets received by a GPCR, which activates a G protein, which sets off a phosphorylation cascade and activates a protein known as Fuse3. Fuse3 binds to the cell membrane and activates a second protein or enzyme known as formin. Formin initiates the growth of microfilaments so that we keep building more and more and more microfilaments to push the cell membrane out to create that shmoo projection. Okay, so this is a great signal transduction pathway leading to the response of changing the shape of the cell. And the reason we know that it's this pathway that plays a role is because, check this out, if there's a mutation that makes formin not work, the shmoo never forms, right? Because we need the formin to build it. But if there's a mutation that knocks out Fuse3, it still doesn't build, even though it has functioning formin, because the message has to be passed from the phosphorylation cascade over to the formin by Fuse3. So if you knock out Fuse3, you prevent this whole thing from happening and you get a bunch of cells that can't make shmoos. It's also just fun to say shmoos. Okay. So let's look at how we can fine-tune these three steps: reception, transduction, and response. There's kind of 4 basic ways to do this. Amplifying the signal, which amplifies the response. We saw one example of that looking at epinephrine and glucose production. Specificity of a response, the efficiency of the response, and how we terminate the signal. So let's look at each one. We know that having an enzyme cascade, like we saw in the epinephrine to glucose cascade, will amplify the cell's response because at each step, the number of activated products is greater than the preceding step. That's the example we already saw. Then we have the specificity of the cell signaling. So you all know that different kinds of cells have different collections of proteins, and those different proteins allow cells to detect and respond to different signals. Now, what's interesting is the same signal can have different effects in the cell if the cell has different proteins and pathways. We can even see phenomena like branching and crosstalk to help modify the signal. So let me show you what I mean by that. Here we have the same signaling molecule shown in each of these images. And in one case, it binds to a receptor, sets off the relay molecules, and triggers one response, response 1. In this case, the signaling molecule in the second spot binds, sets off a response that actually causes branching. This activated molecule activates something that's going to trigger response 2. This one activates something that'll trigger response 3. That's branching. Crosstalk, interestingly enough, is when, again, you have a pathway going straight down from one signal, but then some other signal and receptor can either further activate the process or inhibit it because what's happening in this relay is talking to what's happening in this relay. And then finally, even though we have the same signaling molecule, if you have a different receptor, it will trigger a different response. So compare what you see in the first box, response 1, to what you see in the second box, response 5. Same signaling molecule, but different receptor. We're going to see an example of this later when we talk about hormones, when we look at the effects of epinephrine on blood vessels in the digestive system versus blood vessels in your muscles. Okay. Next up for fine-tuning the response, we have signaling efficiency, and this is often achieved by using what are called scaffolding proteins. These are large proteins that basically hold the other relay proteins together so that they're more efficient. They're grouped together so that they can work very quickly to each other. Sometimes they can even help activate the relay points. So for example, here's a big scaffolding protein that's holding 3 kinases, and it can actually hook on to the receptor to help make the process go more efficiently. And then finally, we have termination of the signal. So it's really important to be able to stop the cell signaling from continuing, to basically inactivate it. And usually this is achieved because ligand concentration will fall, which means that fewer receptors are bound up, so less transduction is occurring, okay? Because those unbound receptors revert back to their inactive state. So basically, signal transduction pathways will keep going as long as there's ligand, but as the ligand concentration falls, they'll stop. Now, an interesting phenomenon that is the result of signal transduction is apoptosis. I used to think it was apoptosis. I was incorrect. It is apoptosis. The second P is silent, like in pterodactyl, like the P in pterodactyl. So apoptosis is basically programmed cell death. It's where the parts of the cell get chopped up and packaged into vesicles that can be digested by scavenger cells. This is really important because, as you know, there are enzymes inside of cells, like in the lysosomes, that could damage neighboring cells if they leaked out, right? They would digest them, basically. So doing this programmed cell death allows you to break down the cell, but keep it contained so it can be disposed of without causing damage. This is very different from cell death that is caused by trauma or damage. So here we have an example. On the right-hand side here, you can see a cell that's undergoing apoptosis. It's forming these bumps, these vesicular packages here called blebs. And this, I believe, is a white blood cell that's going to come basically eat it and digest it. So apoptosis is really important for cell development and organismal development in a lot of ways. It helps determine the shape of the organism. For example, your hands during embryonic development have cells in between all of the digits. They almost look like little paddles. But in order to be born with individual fingers, that tissue in between the digits has to die. So that's apoptosis. It gets rid of those extra cells that we eventually don't need. Now, one organism where there's been a lot of studying, actually a lot of studying across a lot of different topics, but particularly that we're going to talk about today is with apoptosis, is called Caenorhabditis elegans, or C. elegans, which is a small worm. Now, we see apoptosis happening when proteins that accelerate apoptosis override those that are putting the brakes on it. So basically, there are always proteins in place that are saying, do cell death, and then there are always proteins in place saying, no cell death, no cell death. And it's just a question of like which way the balance goes. More towards accelerating the death or more towards preventing the death. So let's look at that. In this first cell, you'll notice that there's a receptor for the death signaling molecule, but there's no signal in it. So we have this active protein called SED9. SED9 inhibits the activity of a protein that will trigger a death process. It's called SED4. Okay? So as long as there's no death signaling molecule, SED9 is overriding SED4. But once a death signaling molecule comes in, it inactivates SED9. Now there's nothing stopping SED4. SED4 becomes active. It activates SED3, and they activate proteases and nucleases. These are enzymes that digest proteins and DNA, things like that. So the cell will start to break down and form these blebs that we talked about. So again, what we're looking at here is there's an active inhibitor that's turned on all the time. Unless a death signaling molecule comes, it turns off the inhibitor. Now the death proteins are allowed to proceed. So what can trigger apoptosis? Well, sometimes it can be that extracellular death signaling ligand like we just looked at in our C. elegans example. It can also be because of bad damage to the DNA in the nucleus, or if there's a significant amount of protein misfolding in the endoplasmic reticulum. Protein misfolding is associated with a lot of diseases, so often those cells will basically commit suicide because things aren't working correctly. And the groups of enzymes that often accomplish this are called caspases. They're the main proteases that carry out apoptosis. So again, if we look back here, a lot of the proteases in this group here, at the very end of the cascade, those are going to be caspases. So we know that apoptosis is essential for development and maintenance. For example, often a cell that has DNA damage that could potentially become cancerous will undergo apoptosis, preventing that from happening. We also know that it's involved in some diseases, for example, Parkinson's and Alzheimer's. We know that if cells are not allowed to undergo apoptosis, that can contribute to some kinds of cancer because you can get uncontrolled cell growth instead of removing cells that shouldn't be there anymore. So this is that example that I mentioned about space between the digits. Initially, when limbs are formed that have fingers or toes or the non-human animal equivalent of those things, there's tissue in between the digits. But that tissue in the digits is going to undergo apoptosis to eventually lead to space between the digits. So you have distinct fingers or toes. And that's it for this lecture.